Hi students, welcome to topic 4. Topic 4 is focused on microbial growth. Here I'm showing the intended learning outcomes for topic 4. So here we want to define the growth of microorganisms. We also need to explain the 4 growth phase in a batch culture. Alright. We also want to know how to calculate generation time for microbial growth, as well as calculating the microbial population in a batch culture itself. In this topic, you also need to know how to describe what is a batch, fed batch, and as well as a continuous culture, as well as the various techniques in isolating microorganisms. Right. We also will talk about the various direct and indirect method for measuring microbial growth. Because we also want to explain the effects of the physical environmental factors on the growth of microorganisms. And finally, we will want to classify the different microbes based on their growth requirements, such as temperature, gaseous atmosphere, pH, moisture content, water activity, osmotic pressure, and the salt concentration of the environment. So when it comes to microbial growth, when microbes are given the required nutrients, they become metabolically active and grow. Right, growth can take place on 2 levels. When in the first level, cells synthesize new cell components and they increase in their size. After that, the number of cells in the population itself increases rapidly. Right, bacterial cells themselves, they multiply by cell division. And although some bacteria can grow by budding and forming hyphae, such as what we find in fungi species, most of the bacteria themselves, they reproduce by this process called binary fission. Let's take a look at what is binary fission. So bacteria reproduce by a process called binary fission. This is a 5-step process. In the first step here, we have DNA chromosome duplication. This is where your prokaryotic chromosome in the bacteria begins to replicate. Replication of DNA in both directions here. So here in this image here, you can see that this is the origin of replication. Okay, the prokaryote have a single circular chromosome, and that's where they start making another copy. So after DNA chromosome duplication, okay, the cell becomes elongate in length, and this is where the proteins involved helps the chromosome to actually break and also move towards the midpoint of the cell itself. This process is called cell elongation. Okay, after which the migration of their chromosomes or the bacterial chromosome will tend to move away from each other to the opposite end of each cells here. Okay, and then eventually the proteins involved in the whole binary fission called the Fitz proteins itself, they form a ring, all right, around the periphery, and then causing a cleavage furrow, as you can see here. So eventually here you have a septum formation, right, that divides the cell, and that's where the plasma membrane, the cell wall material, start to accumulate between the two incoming microbes here. And then when you have a septum being completed, the cell pinches into two, forming two daughter cells here. So here we are looking at the binary fission to be in completion. So binary fission is a very common form of reproduction in prokaryotes and in some of the single cell eukaryotes as well, such as the amoeba and also in paramecium. So let's define microbial growth. Microbial growth is defined as the increase in cell number, cell density, or biomass in a microbial population over time. Okay, so whenever there is a microbial growth, there will be an increase in biomass. The biomass is defined as the mass of the living biological organisms. Microbes are usually cultured in a batch culture, all right, also known as a closed system. All right, the closed system of this microbial growth is where we add nutrients in the beginning, but after which it's closed. Right, we don't add additional nutrients during the cultivation steps. All right, in a closed culture, the nutrients concentration will start to decline over time, and the concentration of the waste will increase over time also. These are the metabolic waste that we are talking about that is produced by the microbes here. In terms of microbial growth in a population, the population growth itself is studied by analyzing a growth curve of the closed microbial culture. So there are typically 4 phases of growth in a closed culture system: the lag phase, the log phase, or also known as the exponential phase. All right, you also have the stationary phase as well as the death or the decline phase. Let's look at individually how this each of the phases can be seen. So as mentioned, microbial growth can be classified into four different phases in a population. The microbial growth itself, you can plot it as the logarithm of the number of viable cells in the y-axis, right, versus the incubation time in the x-axis here. So here you see the growth curve of a closed culture of a bacteria. In the beginning, there's a lag phase. Okay, as it grows in the culture, right, the cell density start to increase, and increasingly this is over time of the incubation. This phase is known as the log phase, also known as the exponential phase, right? Eventually they will reach a stationary phase, right, and then followed by a death phase or decline phase, where the number of cell or the cell density decreases. So let's look into more detail into each of these phases. So let's look into some of the factors first to consider in each phases of the growth curve. So we want to look at the level of nutrients available. In different phases, the level of nutrients will differ. Right in the beginning, in the closed batch culture system, the level of nutrients will be very high. Okay, and then over time it becomes lower in terms of concentration. We also want to look into the amount of physical space available. Right, asking the question: is there sufficient space or not? For the microbes to grow, or is there overcrowding or not? Because over time we know that the microbial growth will increase in size, all right, and eventually this will take up all the physical space available for them to grow. Okay, in the third factor, right, we want to consider the level of toxic metabolic waste that's being produced. The metabolic waste being produced by the growing cells will increase over time as well. All right, this is also true for your cell density, because over time your cell density will increase over time, especially we are looking at total cell density. But also we also want to look into the live cell density. What is the degree of the live cells that are increasing? Because you will need to compare this eventually to how much dead cells are you having also. Okay, live cells and dead cells, they contribute to the total cell density that we can calculate here. Let's have a look at the lag phase first. Right, in the lag phase, this is the initial phase where there is little or no increment in cell density. Right, the cells or the bacterial cells that have been added, okay, are still adapting to the new growth condition. Right, the bacteria themselves, they need to synthesize necessary enzymes to carry out their own metabolic processes. Okay, you will find that most of the cells here are still not dividing yet, but increasingly they will, all right, as they move to the next phase. So in this lag phase, what you will notice that the nutrient levels are high, physical space is high, waste is very low, the cell density is also low. Let's look at the log phase now, or also known as the exponential phase. Okay, so in the log phase, right, this is where you can expect exponential growth rate. The cell density doubles every generation. We're going to talk about generation rate later, so no worries here. All right, so all the cells are growing at the fastest rate possible with the shortest generation time. Okay, here we have the optimal growth condition. You have sufficient nutrients, there's no overcrowding, and what you will see if you plot in a log graph, it will be a straight line. Okay, relatively, if you plot it with the log cell, all right, versus incubation time, this will be a straight line as well. So in the log phase, okay, the nutrients are optimal, all right, the physical space is also optimal, the metabolic waste start to increase, okay, as well as the cell density also start to increase. And typically we will use the log phase or the exponential phase here to obtain the healthiest microbes that we want for our experiments. In microbes here in the case of the bacterial cells. Let's look at the stationary phase now. Okay, stationary phase is where your cell growth will be limited by the availability of nutrients and also the increasing accumulation of your toxic metabolic waste, as well as your physical space. This is a phase where your growth rate equals to the death rate. Alright, the cell density remains constant. Alright, cells prepare for competition and also the harsh condition ahead. Some may even form spores, endospores, or cysts. And also some will even produce secondary metabolites towards antibiotics, just in case if there's any harsh conditions that can come to expect for these microbes themselves. So some do produce this secondary metabolites to counter antibiotics. So in the stationary phase here, right, it will be also or you also find that it was also known as the plateau phase in some of the textbooks, right? In your reading, right, you will notice that in this phase the nutrients are decreasing, your physical space is also decreasing, metabolic waste is high, okay, but cell density remains constant here. Finally, we have the death phase. Death phase is where the condition becomes overwhelmingly harsh. The nutrients itself are depleted, and in the environment itself, there is a high level of toxic metabolic waste. Okay, often there's also overcrowding due to the excessive population within the culture itself. So here, the death rate will actually surpass the growth rate here, right? And the life cell density continues to decline. So in this phase here, you will notice that the nutrients are very low, probably close to zero, right? Physical space is very low, also very limited. Waste is very high. Okay, cell density differs, but often you will see that the cell density decreases, especially with your life cell density decreases also. And here, the cell density, I'm talking about the total cell density, where your life cell density declines and your death cell density increases. So I want to talk about metabolic growth, and why are we obtaining cells in the log phase, also at the exponential phase? This is because the metabolic rate of the individual cells is at the highest during the log phase itself. This phase, as I mentioned earlier, is often preferred for us to use in the industry and the laboratory for large-scale growth of your microbes and your bacteria and your yeast. This is because they are the healthiest, right? Yeah, they are growing well. There's a lot of nutrients available, right? The physical space is increasing being used, okay, but the metabolic waste is also low at the moment, right? So in this phase here, this is where we will often extract them and then use them for experimental purposes, right? So there's some ways to prolong the exponential phase of the microbial growth population. We can either subculture into a fresh media, all right, or we convert this into a fed batch culture where we add fresh media to dilute out your metabolic waste, but at the same time provide new nutrients. Or we can actually keep it as a continuous culture, keeping it in the log phase continuously. On the right here, you will see some of the bioreactors that is used for bacterial fermentation in vaccine production. So often in this culture itself, all right, this could be a fed batch culture where the cells are kept happy and are always in a high metabolic rate, and i.e. in the log phase itself. So let's look at what's the fed batch culture. The fed batch culture is a culture where you add fresh media into the culture to provide more nutrients and then to dilute out the metabolic waste. This is relative to a continuous culture. A continuous culture is not a closed system. It is an open system where your fresh media containing nutrients is added continuously to the culture itself, while at the same time you remove the spam media and the waste. All right. So in this case here, the volume of the culture, the cell density, the nutrient, and the waste level maintain constantly. It maintain a constant level. So cells are growing and multiplying at a constant rate as well. This is a continuous culture. So now let's take a look at how to calculate generation time for microbial growth. So we know that microbes are like bacteria. They divide, all right, through reproduce through binary fission, and one cell often give rise to the daughter cells, two daughter cells. So each round of the binary fission itself is actually called a generation. Okay, so with each generation, the cell number doubles. The time taken for one generation or for the cell number to double is also known as the generation time or doubling time. Okay, let me repeat that again. The time taken for one generation or for the cell number to double is also known as the generation time or doubling time. Okay, so the generation time itself is specific to the species. Different species have different generation time or doubling time, and it differs also with the growth condition subjected to the species itself as well as its stage of growth. So in the earlier slide, we talk about generation time. Okay, so generation time is the time taken for one generation or the number of cells to double. So in terms of generation time, okay, your initial parental stage can be consisting of one cell, and then in the next generation, the first generation will consist of two cells, right? And then so on in the second generation, you have four cells. The third generation having eight cells, then 16 cells, and then so on and so forth, 16, 32, 64. So as long as the environment is favorable, all right, this doubling effect can continue at a constant rate. The population will double over and over again. So the length of the generation time itself is also the measure of the growth rate of the microbe in the culture. Right, most of the growth rate of the bacteria is very fast. Their average generation time can range from 30 to 60 minutes under optimal conditions. Some as short as 10 minutes is also possible. All right, but for other microbes such as the mycobacterium, right, they are often required in the medical microbiology lab a long culture days. So often you require days for them to grow to sufficient numbers. Hence, their generation time for this species, or in fact this genus itself, Microbacterium, it can last between 10 to 30 days. So we know that the microbial growth itself can be represented by the exponent. So when we talk about exponent number, often the cell population itself is represented by the number 2 with an exponent of either 1, 2, 3 or 4. So the exponent increases by 1 in each generation, as you can see here, and the number of exponent is also the number of generation itself. So exponent 1 is generation 1, exponent 2 is generation 2, exponential 3 generation 3, the third generation, as well as exponent 4, the fourth generation. So because this population often contains very large number of cells, it's very useful for us to express them in terms of exponent, or also known as logarithm. All right, so often when you plot the data, okay, from a growing bacterial population, you plot it based on the number of cells as a function of time. Okay, so the cell number here can be represented through a logarithmic scale here. So plotting the logarithm number here over time will exhibit or show or produce a straight line. So this straight line also is indicative of exponential growth. If you were to plot the data here, okay, using just raw numbers, all right, then you will see that these will often be producing a sharply curved slope here. So it is difficult to assess the number of cells here, all right, just based on the normal raw numbers here. So preferably we often plot logarithm or log graphs here, because we can read out an accurate cell number, especially during the early growth phase. So predicting the number of cells that will arise during the growth phase is quite simple, right? So often you can understand that, with your number of cells here, you will be able to determine how many generations has happened, and we have a formula to derive this. So let's take a look into that. So let's take a look at the equations that we can derive to form a formula to calculate generation time for microbial growth. Okay, so if you were to assume that n naught is the initial cell number and nt is the cell number after time defined as t, all right, and n is the number of generation, then the first equation will be nt equals to n naught times 2 to the power of n. All right, So if you were to assume that G is the generation time, then we can find N, which is the number of generation, to be from the calculation of T, which is time, divided by G, which is generation time. So time divided by generation time will give you the number of generation. So this is the second equation. So if you were to combine equation 1 and 2, so we will get the formula of NT equals to N naught times 2 to the power of T divided by G. So NT is the cell number after time. Okay, N naught is the initial cell number multiplied by 2 to the power of N, where N is equals to time divided by generation time. So please note that this formula is only applicable when the culture is in its log phase. Okay, so if you know any of these two values here, all right, it's easy to find the other values through calculations by making it the subject. So let's take a look at this example. All right, so how many E. coli cells will we have at the end of 18 hours, an overnight culture, if we start the culture with one cell? All right, so let's assume the generation time for this bacteria is around 20 minutes. So have a go at this. Okay, so let me give you the solution to this. So here we are asking how many E. coli cells will we have at the end of 18 hours if we start the culture with one cell, taking the generation time to be 20 minutes. So if you were to use the formula that we talked about earlier, NT equals to N naught times 2 to the power of N, where N is equals to T divided by G. All right, so here we can plug in the numbers. NT is the cell number after time. So this is what we want to find out. So equals to 1. All right, 1 is the starting culture, initial cell number, multiplied by 2, all right, to the power of 18 times 60. Why 18 times 60? So here we're looking at 18 hours as the time. Okay, here we're changing it to minutes. Okay, and then here we are dividing it by 20 with the generation time. G is by 20. So what will be the cell number after 18 hours? So simply this will be 1.8 times 10 to the power of 16 cells. Right, so is it possible or not? If one cell weighs roughly about 10 to the power of minus 12 grams, and we have about 1.8 times 10 to the power of 16 cells, that will be essentially 18 kg, right? This will be crazy, right? So a batch culture does not grow continuously, right? So you want to take note on this. In fact, the growth of a batch culture is not constant. It goes through the 4 phases as we have covered earlier. So that's why we won't expect the theoretical calculations here to be quite accurate, because in fact the growth itself is not constant. The growth is only constant during the log phase, and then it goes through the 3 other phases that we talked about earlier. Okay, let's look at another example of this. This is the experimental data of a yeast culture. After 2 hours, all right, we start off with 5 times 10 to the power of 5 cells. After 4 hours, we end off with 5 times 10 to the power of 6 cells. So the first question to ask is, what is the generation time of this yeast culture? Right. Second question here, can you predict the number of cells present after 6 hours if it started off with 1000 cells, assuming the culture is in the log phase? So please have a go at this, and then I'll show the solution later. Okay, so let's look at the solution for this example. So let's assume that the data collected here are from the log phase. So if we were to use the same formula, NT equals to N naught times 2 to the power of T divided by G. If you were to plug in the numbers here, NT refers to the cell number after time. So this will be 5 times 10 to the power of 6 equals to 5 times 10 to the power of 5, which is for N naught, the initial cell number here, all right, times 2 to the power of 4 minus 2 divided by G. 4 minus 2, 4 is the 4th hour, and then 2 is the 2 hours in the initial phase. So here, if you were to make 2 to the power of 4 minus 2 divided by G as the subject, which will turn off as 2 to the power of 2 divided by g. All right, moving this 5 times 10 to the power of 5 over, right, you eventually get this equation here, which is 2 to the power of 2 divided by g equals to 5 times 10 to the power of 6 divided by 5 times 10 to the power of 5. All right, so after which, if you were to punch in the numbers, this will be the next equation. All right, still making this as a subject, 2 to the power of 2 divided by g equals to 10. So here we will want to apply logarithm to both sides, so that we can remove or in fact remove the log to isolate the variables that are in the exponent here. So in this case, the exponent here will be 2 divided by g, because we were interested to find out what is the generation time, right? And that's in g here. So when we apply log to both sides, all right, log 10, all right, log 10 eventually we know is 1. All right, so this will result on the left hand side here, all right, as 2 to the power of g log 2. All right, why are we seeing 2 to the power of g where your exponent is being brought down? All right, here is because of the property of equality in the rules of logarithm, which states that if two expressions are equal, all right, then their logarithm are also equal. So you might also ask, why are we doing this? The primary reason is because we are using the power rule, right? So which states that, all right, just let me get my my pen out. All right, so which states that the power rule, all right, which is log, okay, x to the power of y, right, is equals to y log x. So hence you're able to bring down your exponent here, which is your 2 to the power of g down, and then having your log state here as well. This is due to the power rule of logarithm. So allowing us to bring down the exponent in this case, so that it's no longer in the power position. All right. So now from this equation here, you can actually move log 2 to the right hand side here, because here this will bring it down to equation of 1 divided by log 2, and then here this will be on the left hand side here, it will be 2 divided by g. Okay, so with this we can now have g as the denominator rather than the exponent. Then moving forward, you can actually do a simple solving by algebra. So let me get my pen out again. So what you can do here, this will be 2 divided by g is equals to 1 divided by log 2, and then if you do the crossing, making g as the subject here as what you see here, then this will be giving you g equals to 2 log 2. So if you punch in this equation 2 log 2 into your calculator, you will get 0.6 hours. So hence the generation time for the yeast culture here is 0.6 hours. Okay. Okay, for the second question, it's asking what is the number, all right, predicted number after 6 hours if we start off with 1000 cells. So from the previous question, we know that the generation time for the yeast culture is 0.6 hours. So if you use the same formula again, nt equals to n naught times 2 to the power of t divided by g. All right, here we are finding out what will be the final number here after x number of times. All right, so how long, or in fact, how many cells will be present after 6 hours? So if we use 1000, that will be your initial start number as given in this question, times 2 to the power of 6, because here we want to find out after 6 hours what is the final cell number here, right? And then divided by the generation time, which we know is 0.6. So if you punch in the number and the equation here in your calculation, in your calculator, sorry, then after 6 hours there will be 10 to the power of 6 cells as derived here. Okay, so let's switch gear for a bit. Let's talk about some of the techniques that we can use to isolate microorganisms, isolation techniques. So in microbial growth, it's important that we isolate bacteria and other microbes so that we can properly identify them. Because after identification, the appropriate antibiotics, for example, can be used to treat infection caused by this bacteria, be it whether is it gram positive or gram negative. Okay, the identification of bacteria is also important as we track and control epidemics, and particularly in some of the water industry, we actually isolate strains of the bacteria and try to quantify the amount of them, and hence we need isolation techniques in order to help us to do this. However, it's quite difficult to isolate them because bacteria exist in a community with other bacteria as well. And isolating a single specimen, or in fact, single species itself in the first step to identify is quite difficult. And hence, learning the appropriate isolation techniques can help us to successfully separate them from other species, right? In addition, we will also need we also need the knowledge of their preference in terms of their optimal temperature for growth, their optimal oxygen requirements, as well as their optimal nutritional needs. So generally, there are three ways you can isolate microorganisms. Firstly, we talk about strict plate, the pore plate, as well as the spread plate. So by now, you should have actually done the strict plate, and probably we are in the process of going towards doing the pore plate as well. Some of you may have already done it before. Alright, these are the generally three ways you can isolate microorganisms. And this method can often be done at the working bench in the laboratory. In our laboratory at TP here, ASC, most of our microorganisms we are using are non-pathogenic strains, also known as BSL1, Biosafety Level 1 microorganisms. In the industry, particularly in the clinical microbiology lab, you may encounter microorganisms that are pathogenic and hence a higher BSL level, and this could be in BSL 2 or 3. And the higher you go up to 4, it's more important for the laboratory to have containment strategies to ensure that these microorganisms do not escape or cause ill health to the users in the laboratory. So I want to talk about the first method in terms of isolation technique. So this is the strict plate method. The strict plate method is a very rapid dilution technique that involves spreading your inoculation loop filled with your culture over the surface of an agar plate. This can be a differential media or it can be a general media. So all in all, the main aim is to actually spread the bacterial load with each streak move here. So you can see the different areas of the quadrant, 1, 2, 3, and 4. As you streak over the quadrants, all right, the community of cells are being spread out, apart, further and further away from each other, so that you actually utilize the whole plate. And when they're far apart, you get isolated colonies towards the end. So the 4 ways, or the quadrant streak as they call, is most commonly practiced. Now of course, as you have done this already, you will know that the aseptic techniques are important to perform the method, because you want to prevent any unintended microbes from growing here. And here you can see that we are streaking primarily at the tail end of each of these quadrant of the area, so that you are effectively diluting out the microbes in order to get isolated colonies in the end. Okay, another isolation technique will be the pore plate method. The pore plate method is quite simple. Okay, basically you are getting your bacteria mixed together with warm agar. So that's around 45 to 50 degrees. And you'll be doing this in the laboratory as part of your practical. So as you have this bacterial inoculum, all right, you can pour your media in, all right, this molten agar, all right, and then swirl it gently to mix, and then you can subsequently allow it to solidify before you incubate upside down with your agar upside down when you do the incubation. So after which, probably depending on the species itself, you will see colonies, and some of these colonies will be growing within the agar as well as some on top of the agar. So once again, aseptic techniques is important to be adhered to where you perform the method. And it's also important to know that your sample is diluted sufficiently first before you add it to the molten cooled agar, and pouring it in a petri dish itself, because the isolated cells itself will give individual colonies growing the agar itself. So this technique requires some experience, but with practice you should be able to do it. Mainly the primary concern is if the agar is not cooled sufficiently, it's still very hot, it can actually kill off your intended microbes that you plan to isolate. So another technique to isolate microorganism is the spread plate method. Okay, this spread plate method we didn't manage to do it or practice during our practical, but nonetheless, it's a method that can be used to isolate your colonies. So in this case here, you have a bacterium sample as an inoculum. All right, you are spreading it or adding it as or inoculate it into a solid agar here. Then by using a hockey stick like this, this is also known as a glass spreader. Okay, you use it to spread evenly, spread the bacterial sample solution or the culture over the surface of the agar using the spreader. All right, then the plate is incubated upside down. All right, and then the bacterial colonies will actually grow on the surface on top of the media itself. So once again, aseptic technique is important here. All right, and then you need to make sure that the liquid sample is diluted sufficiently, so that your bacteria are easy to count and isolate also. So often it's desirable to have a countable number, as we casually mention this, right? But the number here, the magic number here is 25 to 250 colonies of isolated colonies evenly distributed on the plate itself, right? So you might ask also, why is it important that we actually dilute the liquid sample sufficiently? And this you will do it in your practical as part of serial dilution, because if you have a too high number of colonies in your petri dish, in your agar plate, then it will be difficult to enumerate them or to count them, making the calculations of the original cell density, or even the what's the colony forming unit per mole concentration of your specimen difficult to assess. So therefore, often we will dilute your bacterial culture in many folds. It can be 10 times, 20 times, all the way to 100 times or even more, so that we can have a countable number of between 25 to 250 colonies that will allow us to have statistical relevant accuracy to determine the cell density of your sample. Okay, in summary, we have looked at defining microorganism growth. This is the increase in cell number, cell density, or biomass in a microbial population over time. All right, we also explained the four growth phases in the batch culture. All right, the four phases are your lag phase, your log phase, your stationary phase, as well as your death phase. All right, it's important to know each of these phases in terms of its nutrient level, its physical space, and also the waste level, as well as the cell density in each of these phases. All right, we also look at how to calculate generation time for microbial growth, and also able to calculate the microbial cell population in the batch culture itself. Remember, batch culture are often a closed systems. So here we are using the formula here, nt equals to n naught times 2 to the power of t divided by g. All right, so nt, this will be your final cell number. Okay, n naught will be your initial cell number. All right, multiplied by 2 divided by time. Okay, this could be in hours or in minutes. All right, and then g will be the generation time. Okay, so we also look at what is batch and fed batch culture and continuous culture. Okay, batch cultures are usually closed system. Fed batch culture can be a semi-open system, but there's nutrients being added in, and so you can have a dilution of your metabolic waste as well. Okay, the continuous culture is an open system where nutrients are constantly added, and then your metabolic waste are being removed continuously as well, so you can maintain your or to propagate your bacterial culture continuously. Okay, so we also look at isolation techniques to isolate microorganisms. It's important to isolate them so we can accurately identify them, so they can help to guide treatment, and also to actually understand their characteristics as well. All right, it's difficult to isolate them, but nonetheless, using methods like this, strip plate, pour plate, spread plate, we can actually isolate them in order to study them. So especially with the pour plate and spread plate, we also talk about why is it important to dilute your cells your bacterial cells in this case, in order to get sufficient number of or in fact appropriate number of colonies on your solid agar cultures or media, in order for us to derive the cell density of the bacterial specimen here. So here we are looking at a magic number of 25 to 250 colonies on the agar itself to give us statistical accurate results. So this concludes part 1 of topic 4. I will see you in part 2. Thank you. Hi students, welcome to part 2 of topic 4. There's a total of 2 parts for this topic, right? This will be the last part for topic 4, so let's jump into it. Okay, in part 2 of this topic 4 e-videos or e-lectures, all right, we are going to focus on describing various the direct and indirect methods for measuring microbial growth. Okay, we will also look through what are the effects of the physical environmental factors that can affect the growth of microbes, as well as classifying them based on their growth requirements here. All right, so let's move into some of the direct and indirect methods for measuring microbial growth. So why do we need to measure microbial growth? All right, so it's natural because we want to study the time progression of microbe products formation relative to cell growth and nutritional utilization. We know that there are 4 phases of microbial growth, and each of these phases it cycles through in a culture itself. So to know exactly how much or how many bacteria are we working on when we start the inoculum and subsequently when doing a subculture. Okay, and importantly also we want to enumerate or even count how much bacteria is it present in contaminated food, as we know that with time the production of toxin from these bacteria, especially if they are pathogenic, is also relative to growth. Okay, so some of the reasons are outlined here are the reasons why we need to measure microbial growth. There are various direct and indirect methods for measuring microbial growth. Some of the criteria for selecting a method, the criteria to consider before selecting a counting method includes firstly, what's the expected microbial load of the sample, or whether the expected count is low or high. The next factor to consider is actually how long or how much time you need in order to get the results from that counting method itself. And finally, whether the method itself involves the use of appropriate growth media for the microorganism of your interest. So the direct methods and indirect methods for measuring growth, most of the time the direct methods measure the cell number directly. Direct methods measure the cell number directly. The indirect methods, they measure the cell other cell growth based on other physical or chemical properties in the culture. So this may include the cell mass. Right, for example, we might be looking at the dry and the wet weight, the nitrogen content, the carbon content, so on and so forth. We might be looking at the carbon dioxide production, evolution of it from the culture, as well as the metabolic activities, probably the generation of ATP from the cells that's being cultured. So all these are indirect methods. All right, the direct methods relatively will measure the cell number directly. Okay, so let's look at some of the direct methods for microbial growth measurement. Okay, so here the first method will be the direct microscope count. We also have the plate count method. Typically, this is done using pour or spread plates. All right, pour plate. We will have practiced this in our practical, and some of you might have already gone through how to perform this plate count method. All right, another method will be the membrane filtration method. Okay, and this is usually followed with the plate count as well as part of the direct method. Let's look into more detail about the individual direct method that we talked about in the last slide. So direct microscope count is one of the direct methods for measuring microbial growth. Okay, this method involves counting the number of cells present in a volume of the diluted culture itself, and often we express it in cells per ml. You can do this count using a hemocytometer, as you can see on the right here as a representative image. All right, this is fast. Typically, we don't need much incubation, but it can be quite tedious and tiring, especially if the cells are moving around and or they are being clumped together. All right. There are also errors that could happen because this is largely dependent on the operator who is counting it under the light microscope. All right, and often it's quite insensitive, requiring at least 10 to the power of 6 cells per mil to give you an accurate readout. So among the many direct methods for measuring microbial growth, this technique, also known as the plate count method, it requires incubation following serial dilution. So typically we will do a pour plate or spread plate so that we can isolate colonies on the solid agar surface. All right, but before that, as you have recalled from the last part of this e-lecture, right, but often the bacterial culture has to be serially diluted to the appropriate concentration, right? So in the pour plate method, typically we will mix it with molten agar with the bacterial inoculum, right? And then when they are incubated, okay, the colonies will form. They are either embedded on the agar itself or on the surface. Then we could remove a single colony of the microorganisms that we are interested in. All right. You can also do this with the spread plate method. Okay. But after which, once you have colonies on your solid agars, you can actually do a plate count method. Okay. To derive the cell density in terms of CFU per mil. So CFU here we're looking at colony forming units per mil, right? So when you do dilution, the serial dilution, and you might have done this during your practical, all right, we are diluting your bacterial culture tenfolds in every of the instance here, giving you a dilution factor of 10 to the power 1 all the way to 10 to the power 6, all right? And then if you're to isolate the individual tubes here, your dilution tubes here, using and to grow them and isolate them using either a pour plate or a spread plate method, then after incubation you will be able to identify and observe colonies growing on them, right? So remember the magic number that we talked about in the early part of our video. We're looking at 25 to 250 colonies, all right? If it's too high, too many colonies, more than 250, we call it TMTC, too numerous to count, too many to count, all right? If it's too low, too few to count, right? Less than 25, all right? Then it will be not significant, to give you accurate results. So the sweet spot is between 25 to 250 colonies, and then in order to derive the cell density, often we will use this formula here. We will take the average CFU multiplied by the dilution factor divided by the inoculum here, right? So in this case here, in this example on the right, okay, we are having probably triplicates being done in each of these instances here in terms of whether it's a pour plate or spread plate. These are triplicates, all right, of the plates, and then you count the average colonies obtained throughout these three plates to derive this number for the average colonies, all right, and multiply by the dilution factor appropriately. This could be 10 to the power 4, all right, and then divided by the volume of inoculum. And inoculum here, we're looking at 1 mil here, all right. So with this plate count method, you're able to enumerate the amount of bacteria in your sample that you have taken in the inoculum, right? So the plate count is most common method to find out the number of live or viable bacterial cells, right? Good sensitivity, you don't need a lot of complicated instruments or equipment, okay? Often this is used for environmental and food sample enumeration of bacteria, but you can see that this is quite tedious and slow lah. And of course the accuracy depends on counting a large number of CFUs within your culture plate itself. All right. Okay, so another direct method for measuring microbial growth is membrane filtration. Okay, membrane filtration is used when your sample has low cell density. Okay, like for example, the fecal bacteria that may be present in a river body, right? and often we will want to use this membrane filtration to concentrate the bacteria for counting. All right. Well, this is unexpected because the amount of bacteria that you sample from a big water body, for example, they are usually very small. All right. So here we are using a large volume of the liquid. In this case, this could be water samples taken from the reservoir and passing it through a membrane here. All right, and the cells will be concentrated and trapped on the membrane here, okay, as the water pass through by size exclusion. Okay, the membrane is then placed onto a suitable agar media, okay, and then the cells will grow into colonies which can then be counted. All right, so in this case here, the number of colonies will be equal to the number of CFUs in the original sample that's taken from the reservoir. All right, so I've outlined a representative flow here for membrane filtration. So often this is done through a 0.45 micrometer membrane filter, right? So the bacteria will be trapped on the filter. The filter is taken out from the filtration unit. A filtration unit looks something like this on the right. All right. Then once you have this membrane, you can apply it and place it on a suitable agar. This could be a nutrient broth initially, and then the cells will grow and after incubation, and then you can count them after which. Some of the advantages and application of this membrane filtration method for direct measuring microbial growth will be for samples with low cell density. As I mentioned just now, you can process a large volume of samples at one time. It's really good for picking up microbial contaminants in any of the samples, not only in the food industry or even in the environmental industry. In fact, in the cellular therapy space, we do use membrane filtration by sampling a small quantity of stem cells from the stem cells collected from the donor to assess sterility. So this is also known as sterility testing to ensure that the stem cell products that's collected from the donor is contaminant free before we give it to a patient for treatment, right? However, this method can be expensive and may be tedious at times. You may come across difficulty or challenges in growing a sufficient number of bacteria, or in fact the media may not be suitable. Okay, so if it's not necessary to count microorganisms to estimate population size or cell density, right, then we can use indirect method that measures variables such as turbidity, dry and wet weight, the nitrogen content, the cell metabolic activities, as well as some of the rapid methods that we can talk about later as indirect methods for measuring microbial growth. So let's go into individual methods here in more detail. Okay, so let's look at the first method, the first indirect method. This will be turbidity measurement. So we know that microbial growth in liquid broth, for example, nutrient broth, as microbe grow in numbers over time, the liquid media or liquid broth will appear to be turbid. So the higher the cell density, the more turbid the broth will be. So often we can use a equipment known as spectrophotometer to measure the turbidity. A spectrophotometer is shown here on the right here in the representative image. So this is where you place a sample in and then you have a beam of light passing through the liquid broth and it reaches a sensor detector towards the end. So the light will scatter such that when there's a more turbid solution that has been encountered, you have a lesser light reaching the sensor detector, and hence this increases the optical density, OD for short. So typically for bacteria, we measure bacterial culture using optical density at 600 nanometers, and the higher the OD, all right, the more turbid the sample is. Turbidity measurement is easy and quick. All right, often in the research environment, the research laboratory, when we grow microbial cultures, we often use turbidity as a way of measuring growth. Because turbidity can also be correlated to the plate counts if they are grown under identical growth condition, allowing us to extrapolate to cell density. Okay, however, there's a drawback. All right, the turbidity measurement does not measure either dead cells or living cells individually. Yeah. Okay, so this means that both dead and living cells contribute to the turbidity. So if you want to assess living cells, the viable cells, you need to use another method. Okay, the turbidity measurement may also be not very sensitive. Okay, it requires at least 1 million cells per mil to give you an accurate readout from there. Okay, the second method for indirect method for measuring microbial growth is also using the dry and wet weight measurement. So for wet weight, usually the aim is to separate the cells from the growth media by filtration or centrifugation. Then we remove the supernatant, which is the resultant top part, top liquid portion of the whole cell specimen. Okay, then we will want to weigh the packed cell mass itself, which is at the bottom of the specimen itself. Okay, so the packed cell mass itself typically is proportional to the number of cells in the culture. All right, that's for the wet weight. All right, for dry weight, typically we will dry the cells in the conditions here and then we weigh them. Okay, but the dry and the wet weight is fairly insensitive. It requires certain levels or certain number of cells, and it may be difficult to weigh the small amounts that's been obtained towards the end. So usually this method is used for filamentous microorganisms such as some of the fungal species. Apart from the last two methods, for indirect methods for measuring microbial growth, we can also determine the nitrogen content of the microbial culture. So here we are measuring the amount of organic nitrogen in the cell mass, and we have a method to do this using the Kajalha method. So as more and more cells make more proteins, right, the higher organic nitrogen content will result. So in this case, you just need to know what is the method that's used to measure the nitrogen content in the microbial culture. All right. So another indirect method will be looking at the metabolic activities in the culture. So we know that microbial growth is characterized by increased metabolic activities. So we can measure the substrate consumption, right? The metabolic waste that's been accumulated, like for example, carbon dioxide, or even some of the acids being produced, because these are all directly proportional to the bacterial number in the culture. So I want to talk about some of the rapid methods that are often used in the research laboratory. These methods can be fast and sensitive, but can be expensive to carry out. And there's also another caveat of using these rapid methods, because some of the results here are obtained from these methods can be difficult to correlate to actual cell numbers. But nonetheless, it provides a first-line estimate of the cultural growth itself, so that you can quickly determine if your cultural growth is it's growing rapidly, or is it in a different is in a plateau stationary phase where there's a lot of dead cells, for example. Right. Here some of the rapid methods may involve the measurement of ATP. We know ATP is produced as an energy source from microbes, so if you're able to measure the ATP amount, right, that will possibly give you an insight of whether there's a lot of viable cells in the specimen culture. All right, you can also use DNA techniques such as real-time PCR gene probes, all right, to determine the genetic content of the specimen. Okay, we know that certain eukaryotic cells, they are nucleated, so with a higher readout of your genomic DNA, for example, then that will give you a estimate of the cultural growth in your specimen. Okay, so in the last part of this lecture in the topic, I want to explain some of the effects of physical environmental factors that can affect on the growth of the microorganisms. So the growth of microbes is affected by its physical environment. So here we're looking at temperature, pH, gaseous atmosphere, the water availability, osmotic pressure, as well as salt concentration in the environment. Different microbes have different combination of environmental requirement. Okay, so let's have a look at each of these factors. Okay, so it's important to encourage optimal growth of the useful or beneficial microbes. And in order to do that, we can often control the physical environment so that we also inhibit other harmful microbial from growing also that will inadvertently disrupt your isolation or your growth of your intended microbes of interest. So a controlled environment here aims to provide optimal environmental condition for your microbial growth. All right, and here we can control it using such as equipment such as incubators with temperature and or with humidity control. All right, so that we can provide the optimal condition in terms of the temperature to encourage microbial growth. So when it comes to temperature, right, the internal temperature of the cell is often described as the same as the external environment. So the temperature do affect the rate of the metabolic activities that the microorganism carry out. At low temperature, right, as we see in earlier chapters of the topics of this subject, right, we see that if you to refrigerate your cells, all right, at 4 to 10 degrees, often you will have slowed their growth, and this will result in lower rates of your metabolic or your biochemical reactions. Higher temperature tends to denature their proteins, okay, and hence they may not be viable towards the end. Okay, the range of temperature required for the microbes depending on different species, they grow optimally and often fastest at their optimal growth temperature, right? So when we talk about the lowest and highest temperature at which the growth can occur for this microbes, often they are known as the minimum or the maximum growth temperature, right, respectively. So most microbes have only a limited range of their temperature for growth. They have preference, which we will see later on in the later slides. Okay, but some have very wide ranges, like for example Bacillus subtilis. These are between 8 to 53 degrees Celsius. Others, they have a much narrower range, right? Neisseria gonorrhea, which causes the disease of gonorrhea, they work actively at body temperature, right? 30 to 40 degrees. Okay, so we can categorize microbes based on their growth temperature. So here, if you have microbes that are growing optimally between minus 10 degrees Celsius to 20 degrees Celsius, these are typically called psychotrophiles. All right, so they are cold loving. All right, they work best or they grow best between minus 10 to 20 degrees Celsius. They are commonly found in the polar regions. Those are microbes that are psychotrophiles and the deep sea environment. All right. If you have microbes that grow optimally well between 20 degrees Celsius and 45 degrees Celsius, and most of them are typically growing at this range, right? Some of the pathogens and bacteria that falls into this category, such as E. coli, they are known as mesophiles. All right. Moderately temperature loving, if you would like. Okay. You have also thermophiles, which grow optimally at higher temperature between 50 degrees to 60 degrees Celsius. They are very heat loving. All right. They are usually found in hot springs. Okay, and compost bundles of compost. Very high temperature because of all the decomposition happening. All right, and finally you have the hyperthermophiles, which are extremely heat-loving microorganisms that grow optimally between 90 degrees Celsius to 120 degrees Celsius, and often they are found in volcanic environments or even in the hydrothermal vents in some of the deep sea. So another factor that can affect the um, growth of microbes is pH. So pH, as we know, measures the concentration of hydrogen ions in the solution. So as your hydrogen ion concentration increases, your pH actually decreases and becoming more acidic. The inverse is true also when your hydrogen ions decreases, then your pH become higher, so becoming more basic and alkaline. So the acidity and the alkalinity of the environment influence the microbial growth. pH affects enzyme activity, which in turn affects biochemical reactions. It also affects stability of enzyme proteins, right? Because it's important for protein folding and its stability. So if microbes that prefer in fact, most microbes will prefer a narrow range around neutral pH, 6.5 to 7.5. These are known as neutrophiles. All right. Usually around this range. Very few are acidophiles that can grow at a pH of acidic pH around below 4. All right, that's why you see that some of the organic acids are used to preserve food, such as fermented dairy products. Others may prefer a more alkaline range, such as those microbes that are termed as alkalinophiles, operating at up to pH 11.5. So with pH changes, the pH level do changes with microbial growth. Microbes produce metabolic byproducts during their growth, and this may change the pH environment in which they are being cultured in, such that eventually it may inhibit further growth of many of its own growth or other microbes they may be growing together with the intended species. So like for example, lactobacillus produce lactic acid and other organic acid during growth. So if there are certain microbes that are preferring a more acidic pH, probably below pH 4, those microbes are also known as acidophiles, then they will be much happy within the environment itself, and this may actually spur their growth while inhibiting others. I want to talk about gaseous exchange. Gaseous exchange, in terms of the gaseous environment, is important because some of the microbes require oxygen for respiration to generate energy. Other microbes may not need oxygen at all. However, there is also a subset of microbes that can be killed in the presence of oxygen. So the toxicity of oxygen cannot be understated. The gaseous and dissolved molecular oxygen, as we know, they are not toxic. So we're looking at O2 here. However, metabolic reactions that often transform these oxygen into highly reactive forms, all right, so-called forms, okay, may form or may result in the generation of some of these radicals as we come about. So these are superoxide radical, peroxide anion, as well as your hydroxyl radical. Because these highly reactive oxygen species, they will react with macromolecules, right? And they are forming adducts that can potentially destroy these macro molecules. So here we are looking at the reactive oxygen species. Some of these are outlined in the next slide. So as microorganism uses oxygen in their aerobic respiration, right? These reactive oxygen species may form as byproducts, and it's important that the microbe itself contains enzymes to actually detoxify itself. So some of the reactive oxygen species or ROS that we talk about, that includes your hydrogen peroxide as well as your superoxide. Okay, so this is indicated by hydrogen peroxide, H2O2. This is a byproduct. All right, you also have superoxide, which is your O2 minus. Okay, so it's important, as I mentioned, right, that the cell protect itself to defend against these ROS. All right, and because these ROS have the potential to form adducts when formed with bio macromolecules, and this may damage cell components. So typically some of the microbes, they have enzymes such as superoxide dismutase that will neutralize your superoxide here, all right, breaking down into hydrogen peroxide and oxygen. All right, you also have some of the microorganisms. In fact, some of the microorganisms all have the enzyme known as catalase. So catalase will break down the hydrogen peroxide to harmless water and oxygen. All right. An important feature of some of these enzyme is peroxidases. Right. Peroxidases also help to reduce hydrogen peroxide to water using NADH. So in the combination with NADH and hydrogen ions, it will form the NAD metabolite, but also water is also produced in the process also. So the hydroxyl radicals that's generated from your hydrogen peroxide are readily removed. In the presence of these enzymes that that the microorganism may have to protect itself against these ROS species, preventing it from accumulating to damage cell components, which can be detrimental for their growth. So the tolerance to oxygen of the microorganisms depend on the availability of these three enzymes. So strict aerobes and facultative anaerobes, they do possess this enzyme to neutralize this ROS. So when we talk about aerobes, aerobes are strict aerobes. They are microorganisms that require O2 to be present so that they can grow in an aerobic environment. When we talk about facultative anaerobes, right? So they are able to grow, in fact, grow better if oxygen is present. But they can also grow without oxygen, and hence the term facultative, meaning is the microorganism is very flexible. If you give me oxygen, good, I can grow. Without it, I also can grow, no problem. So facultative anaerobes, all right, they use aerobic respiratory respiration actually, if oxygen is available. And if it's not available, then they will switch off to other alternative type of metabolism. All right. So often growth is also faster for them when oxygen is present, because we know that aerobic respiration processes produce the most ATP. Yeah. And interestingly, E. coli is also a facultative anaerobe that's present in our large intestine. Okay, so I also outline another type of species, or in fact microorganisms, that these microorganisms may be aero tolerant anaerobes, because they may also possess some of these enzymes to help defend or help to neutralize this ROS. So when we talk about aero tolerant anaerobes, all right, they are indifferent to oxygen, right? When they can grow in the presence of oxygen, okay, but they do not use it to actually generate energy. Okay, so these are some of your microbes that perform fermentation to actually use it as their energy source. An example of this is Streptococcus pyogenes, which causes sore throat or strep throat. They can grow in the presence of oxygen, but they can choose not to use it to generate energy. Okay, we also have another category of microbes that have limited ability to detoxify oxygen, especially with the ROS. These are microaerophiles. So microaerophiles, as the term shows, microaerophiles, they require very small or micro amounts of oxygen. So typically 2 to 10% for aerobic respiration. Okay, higher concentration is quite inhibitory to their growth, because due to the fact that they have limited ability to detoxify ROS. Depending on the species, they may or may not have all the three enzymes that we talked about in the earlier slide. Okay, so an example of this is Helicobacter pylori, which causes gastric and duodenum ulcers. Okay, so finally we have the last category here, which is the strict anaerobes. So the strict anaerobes, they are strictly requiring anaerobic conditions. That means you cannot put them in an oxygen environment. If you put them in the oxygen environment, they will not grow well, right? Because they do not possess some of these enzymes that we talk about to detoxify reactive oxygen species. Okay, once again, It's important to control the gaseous atmosphere to ensure that the microbes can grow optimally well. So we mentioned about aerobe. Remember that an aerobe is an organism that requires oxygen for growth. It grows in the presence of atmospheric air. An anaerobe is referring to an organism that can only grow in the absence of oxygen. It cannot grow in the presence of our atmospheric air. Okay, so just to recap on what we have mentioned earlier, you have obligate aerobe. Obligate aerobes are aerobes that require oxygen, okay, for growth, for respiration. All right, it cannot grow without oxygen. You have the obligate anaerobe. They do not require oxygen at all, so don't give them. Okay, if you give them, they cannot grow. Okay, you have the facultative anaerobe. So as mentioned earlier, facultative, they're very flexible. All right, you give them oxygen, they can grow. They can also grow without oxygen. Okay, so if oxygen is available, of course they will use it for their energy production, for ATP production through aerobic respiration. Okay, you have another category of microbes that may be microaerophiles, need micro or small amount of oxygen, grow well at 2 to 10%, but if you at higher levels, they are very inhibitory in their growth. All right, you have aerotolerant anaerobes. They do not require oxygen. Okay, they are very indifferent. They can tolerate oxygen even if you give them. Okay, and finally we have the kinetophiles. Okay, the kinetophiles, they grow best at increased carbon dioxide at high levels with reduced oxygen levels. Okay, I want to talk about water and water availability. We know water is important because it's a solvent for nutrients and enzymes in our body. Very important reactant in many of the metabolic reactions that we come to know about. All microorganisms require water to survive, and most would die if you deprive them of water. So some bacteria will even sporulate, while other groups of microorganisms such as algae and protozoa, they form cysts themselves so that they can tie over the very dry or even harsh conditions before water is made available. So another factor of affecting the growth of microorganism is water activity. So water activity relates to water as what we have discussed in the earlier um, slide. Water is important for most of the microbes itself. So, but in this case, we are talking about AW, water activity, term as AW, because AW is a measure of free water that's available to the cell. We're not talking about the total water availability. We're talking about a measure of free water available to the cell. So in general, AW or water activity decreases, all right, when your solute concentration increases. So pure water has an AW of 1. So if you have a sucrose solution at about 66% weight per volume, then this will bring down your AW to less than 0.85. Most bacteria require AW of 0.96 and above to thrive. Although many of the yeast and mold can actually grow at a depth at low AW, less than 0.9. Zero tolerant microbes, all right, they can actually tolerate very low AW, very dry condition. All right, these are also known as tolerant microbes. Less than 0.6 AW, right? Growth is also not possible, mostly not possible for most microbes itself. Why is it important to know the water activity? Right, it's important especially in terms of food preservation. In food preservation, we want to prevent spoilages of food, all right, due to the growth of unintended or pathogenic or spoilage bacteria, and hence knowing the AW here will effectively extend the shelf life of the food, because in food preservation often we increase the solutes like the salts or even sugar or even removing water so that we can inhibit the microbial growth themselves. Okay, I want to talk about osmotic pressure. Osmotic pressure relates to more on osmosis also. So we know that all microbes require water for growth. Even if water is present, it may not be readily available in certain environment, as we see earlier. Okay, so the fact that in many of the environments, all right, let's say sea itself, right? The sea itself has a high solute concentration, all right, and often this will affect the growth of the microbes if they are placed in a certain environment here. So when we look at solutes or salts in a water body or even a solution, often we'll think about osmosis, right? Osmosis is the diffusion of water across a semi-permeable membrane from a region of low salt concentration to a region of higher solute concentration. Okay, so in this case, water will flow from low solute concentration to high solute concentration here. So in along the same line, all right, we will also come across the term of osmotic pressure, right? Osmotic pressure is defined as the pressure exerted on the membrane itself by the solution itself. So effectively, we will come to understand that the effects of different solute concentration on the cell itself can have an impact on the cell growth itself. So here, if a microbe is being placed in a hypertonic solution, okay, that means it is in a very high solute concentration. Okay, they will experience a very high solute concentration outside the cell. All right, and then what will happen is there is a high water potential inside the cell itself. Okay, and with the principle of osmosis, all right, it's the diffusion of water from a lower salt concentration to a region of higher salt concentration. So the water moves out, right, to the region of the higher salt concentration in the hypertonic solution. So what happens to the cell? The cell shrinks, right? Water has been moving out, right, leading to cell shrinkage. Okay, this causes the cytoplasm to dehydrate, okay, to shrink from the cell wall. Okay, this is because the solute is higher in the environment than in the cell. Okay, this phenomenon we call it as plasmolysis. All right. Okay, then we also have a condition where the microbe may be placed in a hypotonic environment. Okay, so in the hypertonic environment, hypotonic, all right, you have a higher salt or solute concentration inside the cell. All right, hypertonic means the solution itself has very little solutes. Okay, as you can see from the illustrations here. Okay, this will result in the diffusion of water, all right, from the external environment to move into the cell. Because remember, the osmosis is the diffusion of water across the membrane from a region of lower salt concentration to a region of higher salt or solute concentration. So in this case, in the hypertonic solution, the region of higher solute concentration is within the cell itself. So there's a higher water potential outside. Water moves into the cell. This causes the cell to swell and may burst. All right. So effectively, there is no cell wall lysis, but the cell actually swells up and may burst. Okay. So the best case scenario is we want the cells to be happy, right? We will place the cell often in an isotonic environment here. So isotonic, often we are operating at 0.9% saline solution. Okay, so this works well for the cell because they have the same solute concentration across the cell membrane. Okay, and there's equal water potential. So the osmosis, there's no net movement as well, and hence the cell will be very happy. It will be in a normal situation here. So we have seen many factors that can affect the growth of microorganisms. And in earlier slides, we see that water activity, AW, is very important, as well as the water availability relates to a high solute environment can affect or impact microbial growth in affecting their cell wall integrity. Yeah? So much of the time, growth inhibiting effects of high salt as well or sugar concentration is quite good for food preservation, as we see. Often like food like bacon, anchovies, often they are preserved in high levels of salt, right? Sugar is also as a preservative, right? They are, especially honey, they have a high sugar content. That's why they have a long shelf life, right? But nonetheless, there are microorganisms that require high salt concentration to grow. Although we say that some of the microbes, they don't really like the high salt concentration, they will inhibit their growth. These are the microbes that can able to withstand high salt concentration, and these are known as halophiles. Halophiles are microbes that can tolerate high salt concentration up to 10%. For example, sodium chloride. They are also known as halotolerant. Okay, some people can tolerate up to 30% or even higher. Halo means salt, so they're very tolerant on the high salt um, environment here. So some of our skin commensal, like Staphylococcus aureus, they can thrive on our skin on the dry and salty environment, because they can tolerate up to 20 or 30% of salt in the environment here. So halophiles, they do um, require a high level of sodium chloride. All right, so many of the marine microbes are actually halophilic. They require about 3% of sodium chloride to grow well. Okay, and some of these halophiles are found in the environment of the deep sea, yeah, or the Dead Sea. In the matter of fact, the Dead Sea, if you have visited the area before, you can essentially float on the water because of its high salt content. Okay, so here's a summary of what we have covered in part 2 of this e-lecture for topic 4. We look into direct and indirect methods for measuring microbial growth. So some of these direct methods include direct microscopy count. This is often done with a hemocytometer with a light microscope. Okay, we also look at plate count methods, often complemented by pour and spread plates. Alright, the plate count method involves counting your bacterial colonies that often fall within the range of 25 to 250 colonies. This is a sweet spot for you to enumerate your original sample cell density. All right, often you will need to dilute your bacterial culture in order to reach a statistical relevant number, and that's 25 to 250 colonies to derive meaningful results. Okay, you will also look into membrane filtration. This is good for processing large volume of solvent or water that contains very low amount of microbial contamination. Okay, we have seen this to be done in the industry that deals with water safety, and also in the field of cellular therapy as well, as part of sterility testing of stem cell products. All right, and often when you have a membrane filtrate from the specimen, you could actually place it on the agar and then allowing it to grow. Colonies grow can then be counted through a plate count. All right. In contrast to direct method, which measures the cell number directly, you have indirect methods that can measure microbial growth. These include turbidity measurement, typically using optical density at 600 nanometer with a spectral photometer. You can also determine the dry and wet rate of certain filamentous microorganisms. All right, the nitrogen content and metabolic activities can also be derived, as often they correlate to the cell mass within the specimen itself. And we also talk about some of the rapid methods that's available, like for example the measurement of ATP or the quantification of genomic DNA in a specimen. Often this provides a first line estimate of your microbial growth in your cultures. Okay, we also look at different factors that can affect the growth of microorganisms. So this includes temperature, pH, gaseous atmosphere, the water activity, osmotic pressure, as well as salt concentration. All right, so this is a good summary for what we have covered so far. All right, so I've included some recommended reading here. Do look out at chapter 9 for the recommended text by Cohen and Smith. This focus on nutrition, microbial nutrition and growth. All right, so this concludes the last part, part 2 of our topic 4. Thank you very much.